I'd like to welcome everyone to the first JNCL, or Juvenile Batten Disease, online presentation from Beyond Batten Disease Foundation. My name is Mary Beth Kaiser, and I'm the Executive Director for the Foundation. Thank you for taking time to join us. I want to encourage you to use the chat feature of the webinar in the lower right-hand corner of your screen to ask questions throughout the presentation. No question is too big or too small. You can post any time, but we will save the questions and answers for after the presentation, and we'll allow as much time as possible to answer all your questions. If you're only able to listen in by telephone, then you can text your questions to 202-812-6462. This webinar is for you, foundations, parents, families, and loved ones of those affected by juvenile Batten disease. I'd like to start by introducing the Beyond Batten Disease Foundation founder, Craig Benson.
Thank you, Mary Beth, and welcome everyone. To our knowledge, this is the first webinar of its kind for JNCL. We hope you'll find the information useful and helpful, and we're prepared to do more. It will be important for us to get your feedback so we can plan for additional webinars. Please let us know what you're interested in hearing more about, and we'll follow up with a survey by email so you can let us know your thoughts. So for about the next 30 minutes, we want to talk a little bit about the goals for this webinar series. We'll also tell you a little bit about Beyond Batten Disease Foundation, where and how we got started, and what our strategy is. We'll talk about some accomplishments, and then more importantly, we'll talk about where we're headed over the next 3 to 5 years. And then at the end of the program, we'll hopefully have plenty of time to answer some questions and any other inquiries that you may have. I guess first I'd like to tell you a little bit about me. First and foremost, I'm Christian and Garland's father, but I'm also the Chief Executive Officer for Myriad RBM. We're a life sciences and diagnostic development company. I'm also, of course, the founder and chairman of the Beyond Batten Disease Foundation. And in addition to that, I'm also a member of the Board of Directors for the Batten Disease Support and Research Association. So BBDF has taken a very comprehensive and strategic approach to driving JNCL research. There's a lot going on, and we wanted to provide an overview of our strategy, how it fits into the bigger picture, and all comes together to create treatments. An important common theme you will hear throughout this presentation is one of collaboration and teamwork. We think it's critical to our success and want to be as transparent and inclusive as possible with our efforts. In that spirit, from the beginning, we've had success with partnerships, and I would like to acknowledge our longtime Texas partners, the Luke and Rachel Batten Foundation, and also want to especially thank Missy and Wayne Herndon for turbocharging our efforts by creating the Will Herndon Fund of the Beyond Batten Disease Foundation. We also really appreciate the great work that the BDSRA has done over the past 25 years and their leadership in all areas of Batten research and support globally. We've enjoyed a close collaboration with them since we started and certainly expect that to continue. And as we go along through this webinar, we will thank and acknowledge many other great organizations that we have excellent relationships with. So let's start with the mission of the Beyond Batten Disease Foundation. Pretty easy and straightforward. We hope to eradicate Batten disease as well as hundreds of other medically devastating orphan diseases. So we plan to accomplish that mission in 2 ways. First, the prevention and diagnosis. We have developed an easy and inexpensive blood test to detect the gene mutations for Batten and hundreds of other rare medically devastating conditions. We'll talk more about that in a moment. And secondly, research and development, which will of course be the main topic for our discussion on this webinar. First, a little bit about the test. We were successful in developing, in conjunction with the National Center for Genome Resources and now the Children's Mercy Hospitals and Clinics in Kansas City, a rare disease genetic test that covers over 600 of these medically devastating rare genetic diseases. The test was beta launched in late 2012 and is currently being used as a diagnostic within the Children's Mercy Hospital network in Kansas City. This particular test has been well published in top scientific journals as well as in mainstream media. It's gotten quite a bit of publicity, and in fact, Time magazine named it in 2012 one of the top 10 medical innovations for the year. We're quite proud of that accomplishment, and we will keep you posted as the test continues its development and when we have more information on the ultimate commercial launches the test as both a diagnostic and a carrier screening test. We'll keep you informed. So let's turn our attention back to our research efforts. We hope you've had a chance to view our animated video, which is a great illustration of the environment we face driving rare disease research and the strategies we'll need to find a treatment and cure. A link was included in your email invitation, and you can also find it on our website, on the Batten Disease Family Association of the UK, BDFA, and on the Batten Disease Support and Research Association, BDSRA, websites. Feel free to use it in your communications. It's a great tool. Our research efforts began against this backdrop. You can imagine discovery on the left with successful clinical trials and treatments on the far right and a growing divide between the two. Our first step was to develop a plan for building a bridge to span the gap. So when we got started, the first thing we did was hire some scientific leadership. We engaged Dr. Danielle Kirkovich because we felt it was necessary to have an objective scientist, in her case a neuroscientist, to help us study Batten disease and to see where we were going. The first thing she did was to review over 1,700 research articles published about Batten disease over the last 40 years. She then compiled all that information into a single database of information, and from there we were able to develop a strategic and comprehensive plan of action all the way from basic discovery to clinical trials. Another key component was to look for ways to upgrade the science by attracting more rock star scientists and ways to increase the amount of funding per project to increase the impact of the work being done. And lastly, we knew we needed strategic collaborations and find ways to work with other partners like the American Academy of Neurology to pool resources and share information, as well as connect and work with like-minded foundations. Many of you have had the privilege of working with Danielle. She has been an important part of our strategy, and for the last year has also been the part-time scientific officer for the BDSRA. That position is now being transitioned over to Dr. Chris Leonard, who will assume the full-time BDSRA scientific responsibilities. So now I'd like to turn the presentation over to Danielle, and she can give you an overview of everything we're doing. I would encourage you to reach out to Danielle with any questions you may have at any time.
Thank you, Craig, and welcome everyone. As Craig mentioned, we would like to share some exciting advancements in JNCL research with you. But first, I'd like to talk about the business of medical research. Before we created a strategy for the successful treatment of JNCL, we needed to know what we were up against, not just scientifically, but also understanding drug discovery and development processes, public policy, who our potential partners are, and how much it will cost to get the job done. So let's talk about the process. The classic approach to treating any disease is to determine what is going wrong in the body and then design drugs to fix or repair the problem. University-based scientists represented on the far left are responsible for discovery science, or figuring out what goes wrong. Once their discoveries are published, the process is then picked up by pharmaceutical companies represented on the right, who walk it down the pipeline shown here to make sure that the drug works like it is supposed to and is safe. In practice, this doesn't happen very often. Published discoveries languish in the middle in what has been called the translation gap or valley of death, because university-based scientists lack the knowledge, tools, and other resources necessary to create new medicines. Pharmaceutical scientists on the far right must choose only to make treatments that can make a profit. When it does happen successfully, and that's rare, it takes over $1.3 billion and 12 years on average to make one drug. Let's be realistic. There are only 20 to 25 new drugs approved each year for all diseases combined. So what's the good news? Actually, there is a lot of good news. Under the Orphan Drug Act, companies that develop rare disease treatments are eligible for grants to offset their costs, tax credits, extra guidance and advice from the U.S. FDA or Federal Drug Administration, expedited review of projects, and a waiver of expensive filing fees. This helps make orphan disease more attractive to pharmaceutical companies and can shorten the time frame for clinical trials and reduce cost. Patient advocacy groups like us, and many of you, our foundation colleagues, have driven medical research to a point where pharmaceutical companies take the handoff. One can create safe and effective treatments in less time as long as we learn from those who have gone before us and don't try to take any shortcuts. To create an overall unified and comprehensive plan that identifies the most promising drug targets to attract pharmaceutical support and give ourselves the best chance for a positive outcome, we studied Entrepreneurial Foundations, who have been the most successful in developing treatments for their diseases. Over 90% of 225,000 medical research foundations fail to reach their treatment goals. We don't want to be one of those. Therefore, we created a proactive strategy backed by the 6 critical elements found behind every single one of those success stories. I'd like to provide a little more detail and examples of how we incorporated these strategies into our plan. The first critical element is to raise the standards of university-based drug target research. Last year, Bayer Pharmaceutical Company revealed that 2/3 of the drug target information they received from university studies is useless for designing treatments and needs to be repeated or thrown out altogether. Amgen, another large biopharmaceutical company, reported an even higher percentage. The valley of death isn't just a break in the pipeline between academic and pharmaceutical scientists. It's a growing cultural and informational divide between academic and pharmaceutical sectors, resulting in billions of wasted dollars, pounds, and euros across all disease categories. To upgrade the quality of university-based drug target identification, funded by Beyond Batten, Batten Disease Support and Research Association, BDSRA, and other participating Batten foundations, we developed a scientific review process that incorporates the 6 critical elements of success. By including quality control elements from the U.S. National Institutes of Health, NIH, the European Research Council, ERC, and the pharmaceutical industry, we are making it possible to develop a treatment. Every single research proposal is now subject to the most rigorous scientific standards at every step of the therapeutic drug discovery pipeline we showed you earlier. Some of the key components include input from experts in Batten disease, objective experts outside Batten disease who specialize in cutting-edge methods and advanced technologies, leading drug development experts, and leaders from the U.S. FDA. Accountability is another important and unique feature we have integrated into our strategy, a shift away from the way funding has been historically managed. Best practices that we have implemented stress accountability, not only in spending, but also by requiring projects to prove their continued relevance to treatment. In other words, not only are we going to make sure that the proposed project gets done in the right way, but we also want to ensure that the project could ultimately lead to a treatment that will help our kids. The second critical element needed for success is to invest in drug discovery. We cannot continue to follow the traditional unsuccessful model of nonprofit and federal research funding. We cannot keep doing what's not working. We cannot keep investing only in drug target discovery, hoping the pharmaceutical industry will notice us. We need to invest in discovering and making drugs too. Let me give you an example of one of our programs that does just that. The Combined CNS Screening Initiative, or CCSI. The CCSI brings together 11 major drug discovery centers around the world to share promising compounds and drug screening technologies and expertise in the search for drugs to treat neurodegenerative diseases like Batten, Alzheimer's, Parkinson's, and others, We have received funding from high-profile foundations and a major pharmaceutical company in addition to the resources provided by the 11 centers. As we mentioned earlier, we cannot stand around and expect to attract the attention of pharmaceutical companies. And we can't stand by and wait 12 years for them to create a drug for another indication to see if it might work for us too. We have to be proactive. We have to have a seat at the table while drugs for neurodegenerative diseases are being developed right now. We've got that seat. The third critical element is to incorporate cutting-edge methods and to create platform technologies. Why is this important or different? The way things are working now in many cases, an individual laboratory working on a very tight budget might develop a tool to be used in Batten disease research. Often, the little amount of money available to develop tools limits their quality and ultimately their ability to advance Batten disease research. Further, the tool remains in the laboratory and isn't shared. That's why it's important for us to create core tools developed to the highest standards that can be made available to everyone. A few specific examples of what we are doing include working with NIH program managers and funded investigators to determine the three-dimensional structure of the CLN3 protein, co-development of specific antibodies to CLN3 protein with antibodies by design, and creating Batten cell and animal models. We have to connect academic JNCL investigators with the best experts, the best technologies, and the best methods. The fourth critical element is the strategic placement of funds to initiate research growth capable of leveraging major funds. As Craig mentioned earlier, we know we can't do this alone. To be successful, our research initiatives must be capable of attracting and leveraging significant third-party funding. Our strategy must be to invest in high-quality, high-impact research that is able to garner support from the U.S. NIH, European Research Commission, pharmaceutical industry, and venture philanthropists. We're going to talk about money in a minute, but before we do, I want to recognize again just a few of our colleagues we've worked with to accomplish this specific critical element. As we talk about the fifth element of success, you may have noticed that many of our projects overlap several of the other critical elements, and this one's not an exception. Access to experts across the continuum, each step from drug target identification in academia on the left, through validation, lead optimization, preclinical development, and clinical trials on the right. So what's important here is what we've been talking about. We need greater integration, interaction, and consultation with experts at every step of the pipeline. Beyond Batten Disease Foundation and our foundation colleagues are making sure we get it right the first time, saving money, time, and hopefully our children. Here are some of the groups who are leveraging and sharing their expertise along all segments of the pipeline. The worst thing that could happen is to have a potentially promising treatment, get a few years down the road, and make common mistakes that send us back to the drawing board. The sixth and most important critical element, partner, partner, partner. At every step of the process, we need to partner with others to achieve milestones, to challenge thinking, and to provide oversight. We need to pull in support from high-profile funders with deep pockets to uncover common drug targets and compounds, and we need to make sure that those who need the information and resources get them. It's not just families and foundations working together. We need to create an environment that encourages, and in some cases requires, collaboration and sharing among research laboratories, industry experts, and pharmaceutical companies. We must work together at every step to exchange information, to support families, and to prevent duplication of funding, so that we maximize every hard-earned dollar, pound, and euro we raise. We should all be proud of the progress made in the last few years. In addition to the ones you've already seen, here are some more success-minded foundations we have worked with or hope to work with in the future. We've now reviewed the 6 critical elements of success and have given you a few examples of how we have integrated best practices learned from other successful disease organizations. Our purpose has been to give you an overview. It would be impossible to cover all of the ongoing activity in one sitting, so we look forward to providing more detail on these and other projects in future webinars. So we promised we would talk a little bit about money. Here's a very brief snapshot. In the last 4 years, Beyond Batten has raised and invested just over $5 million in JNCL research. We've leveraged another $5 million through partnerships with government, pharmaceutical partners, and other large neurodegenerative disease foundations like the Alzheimer's Drug Discovery Foundation and the National Multiple Sclerosis Society. What we are most proud of is joining with you and other Batten disease foundations. We've also been able to leverage indirect support from 2 pharmaceutical companies. While these companies are building upon Beyond Batten funded research to apply our findings to other rare diseases, these efforts actually lay the necessary groundwork for future therapies in Batten disease. To put this in perspective, we have collectively invested $10.1 million. In the same time frame, the US NIH has invested only $6.5 million in JNCL research. You can appreciate that if we are going to sit around and wait for the federal government to solve our problems, we are going to wait a very long time. As you can see, there's been a lot of activity over the last 3 to 4 years. We've made a lot of progress thanks to all of us working together. And even with all that progress, we still have a ways to go because we're only partially to our goal. So let's turn now and talk about strategy moving forward for the next 3 to 5 years. As you may know, our early funding brought world-renowned Italian researcher Andrea Bellabio, his colleague Marco Sardiello, and members of their team to Houston to create a laboratory at Texas Children's Hospital, while other members of their team continued working at the Telethon Institute of Genetics and Medicine in Naples, Italy. Doctors Bellabio, Sardiello, and their associates discovered and published the very first JNCL drug target, TFEB. This was the first JNCL drug target research that was published in the top 1% of 11,000 science and technology journals. This ranking demonstrates the quality of research necessary to meet the demands of the pharmaceutical industry. You can think of TFEB as the master conductor of the house cleaning symphony of the cell. Let me give you a visual of how TFEB works. This is a cartoon of a normal cell with lysosomes, the garbage and recycling centers of the cell shown in white. Normally, when waste material builds up, lysosomes recycle or take the garbage out. This process is damaged in JNCL. JNCL lysosomes work, but they don't work very well. As a result, the damaged lysosomes start to expand to accommodate all of the extra waste material. The cell starts filling up, crowding out other cell functions, and ultimately the cell dies. Investigators have found 3 drugs capable of activating TFEB to enter the nucleus and tell the cell to make more lysosomes. Their research has shown in human cells in a dish and in JNCL animal models that TFEB can clear much of the accumulated waste material. In the animal model, TFEB activation slows the progression of the disease. In short, TFEB seems to prompt cells to take care of their garbage disposal problems themselves. Investigators have found that these 3 compounds are capable of entering the body, crossing the blood-brain barrier, and activating TFEB. As luck would have it, 2 of the compounds are already USA FDA approved for other indications. If we go back to the therapeutic pipeline, that puts us right about here. So how are we going to get from here to a potential treatment? This is a visual representation of our milestone-driven strategic plan to make our best efforts to obtain FDA approval within the next 3 to 5 years. On the left are the steps, across the top are the target dates, and the colored bars represent the amount of time each step takes. Let you take that in for a moment. That's a lot, but we have help. We engaged a group called Orphan Solutions to help us execute this plan. Orphan Solutions is led by Drs. Marlene Hafner and Lisa Beth Furstenberg. Dr. Hafner is best known as the Dean of Orphan Drugs. She administered the U.S. FDA Office of Orphan Products and Development for more than 20 years, establishing not only the groundwork in orphan product regulation for the U.S., but also assisted in the development of orphan product programs in Europe, Japan, and Australia. She is the world's expert on what it takes to design a plan to develop approved treatments for rare disease. And we've already begun supporting much of this activity. Investigators at Texas Children's Hospital are completing preclinical efficacy studies in human cells and animal models. Parts of the Largescale Plan are being funded through the BDSRA grant review process and participating family foundations. In addition, Drs. Hafner and Furstenberg are getting to work organizing clinical teams and designing eventual clinical trials. We're not just focusing on TFEB. Beyond Batten, Orphan Solutions and other groups like NCL Stiftung, Beat Batten, and others are working hard to uncover other drug targets and potential compounds. We know we cannot put all of our eggs in one basket, so we will continue to identify and shepherd the most promising leads down the therapy pipeline. But this plan doesn't come cheap, and I'd like to invite Craig to talk more about that.
Thanks, Danielle. And you're right. Success doesn't come cheaply. I'd like to share with you our estimate of the cost associated with these programs. Now, that's a lot of money and looks very daunting, I realize. But we have had success raising money that we've needed in the past, and we're already implementing significant strategies to raise the necessary funds for the future. I am highly confident we'll get this done. Obviously, the hope and goal when we get down the pipeline is that we have built a bridge that connects all basic discoveries with the tools, experts, and technologies that will lead us to a treatment. And we will have a structure that allows us to continue to funnel promising research across the valley of death until our children no longer have to confront this disease. We have just given a very broad overview for you, and it was really hard to pack 5 years into a 30-minute presentation, much less the next 5 years. We hope to have provided a broad summary of our research activities and our strategy to reach a treatment and a cure. In future webinars, we'd like to delve into more detail on many of the aspects of our plan and on certain projects in particular. We'd very much like to know what you're interested in hearing more about or clarifying anything for you. So now we'll open it up for questions and would love to hear from you. You can use the chat feature at the bottom of your screen to submit your question to the presenter, or you can text your question to 202-812-6462. Again, that number was 202-812-6462.
Right now we're open up for questions. We're going to answer some questions that are coming in from you, but we also had some great questions from last night, and we'll put those in as well. Please don't be shy. Okay, so I'll get started. First question, if TFEB activators are already working in animal models and are already FDA approved, can't we just try it? That's a great question. When something is FDA approved or ERC, European Research Commission approved, that means it's been tested for a particular population, such as children or adults, and for a particular disease that may have absolutely nothing to do with Batten disease. So in this instance, anytime you want to try it for another disease or another population or another indication, you still have to conduct the necessary safety and efficacy trials. So on the one hand, you do have an advantage because the preliminary safety and efficacy trials have already been done during the first indication, but you still have to go through all of the steps necessary to make sure it's truly effective and truly safe for our children. Okay, next question is the end date. I have to apologize, the chart that you saw with the dates, we realized that the tabs are a little bit faint, so it is a little bit difficult. That chart that you saw is anticipated. We anticipate this process taking between 3 and 5 years. And so the dates at the top you see are for 3 years if we started in May, which we have. We've already gotten a lot of activity going on. So that chart, which we will put on our web, we'll send you, we're going to adjust it so it's easier to see. So you'll see that again and be able to refer to it.
But it does go through 2016.
Yes.
And yeah, that's right. We just, I think we mentioned this earlier. We will email a link to this presentation directly to all those who are participating, and then we will also have that available on our website at a later date as well. So this will be, something that you'll be able to share with others to the extent you're interested, or could send them to our website to be able to check it out later.
Okay, next question. How soon can we get started on clinical trials? Anything we can do to go faster? First and foremost, we all appreciate that we all want to go faster and that we all want to work as hard as we can because we know that time is of the essence. Our kids don't have the luxury of time. And so, but unfortunately, we can't skip any steps of the process. And if we rush or cut corners, the experimental data we obtain won't be strong enough to build therapy on. And when we get to the FDA and ask permission to conduct the clinical trials, they won't let us. And what they'll do is say, go back to the drawing board, redo these experiments, redo this. And time again, history tells us that those who have gone ahead of us and rushed to clinical trials have ended up actually extending their time to get to clinical trials because they've had to repeat experiments and do other experiments. And so this is the main reason we've brought in key experts from all along the pipeline at every stage, like Orphan Solutions, to make sure that every step we take is well informed. Next question. What can we do as families and supporters to get to clinical trials? BDSRA, Noah's Hope, Our Promise to Nicholas, Blake's Purpose, and Beyond Batten Disease are supporting an international patient registry to get together with BioMarin Pharmaceuticals that was initially funded and continues to be funded by the European Research Commission. Our boards and the boards of these foundations and other individuals recognize that a well-designed registry is critical for gathering and informing patients and for understanding the natural history of disease. For our case, what we want to be able to do is any treatment we do, we want to be able to compare against the natural history of the disease or what would happen if treatment isn't available. And what that does is it gives us an opportunity to have a control. It's a historical control so that none of the children need to be on a placebo. We don't have to worry about that. So the more information we have in these registries, the better. They're great for identifying biomarkers, ways of measuring treatment efficacy, and any regulatory agency won't let us do a clinical trial unless we have a way of measuring efficacy. So I really urge you to support registries, and also don't worry about being in more registries than one. This is a time when this can be repeated. There are other individuals working really hard on developing registries. Go ahead and sign up for more than one. It's not a problem. Okay. Next question, haven't we been funding research using the scientific merit review process for years? In some ways we have. We've had a dedicated group of really intelligent Batten disease researchers that have evaluated one another and have really pushed science to the point where it is today that we can start talking about potential therapies. And they've done a great job. What is new in our particular process is when you see that pipeline that we showed you that was in brown and beige, that pipeline represents separate steps in the process to get from basic discovery, drug target discovery, all the way into clinical trials. And what we've done that is unique is two things. One, we've brought in experts from every part of that pipeline so that we make sure that every step one is well informed by step two is well informed by steps step 3. So we don't have to go backwards, repeat anything. But the other thing we've done is we've brought in a lot of experts from other neurodegenerative diseases and technology experts. One of the disadvantages that we have as a rare disease is that we have a very small community. But other communities like Alzheimer's and Parkinson's have thousands of investigators and who have spent many years using technologies we haven't really touched yet. So we want their technology expertise. We also want their years of expertise in teasing apart what is specific to the disease and what is a general phenomenon that happens in neurodegenerative disease. So we've really reached out as hard as we can to the top leaders in other disease processes, and what that's also done is it's helped us recruit these key experts and tell them about that disease and say, come study our disease as well. And so we really want to bring in those research rock stars. Okay. Next question, what other drug targets exist? Several investigators are exploring, this is the main focus of Batten disease research for many years, figuring out what goes wrong. You know, step 1 is the drug target, what's going wrong in the cell. And step 2 is to make the drug to fix the problem. And so the main focus of Batten disease research for many years of those 1,700 articles that Craig mentioned are looking at drug targets, what goes wrong in the cell. And we have several dedicated investigators, over 40 labs across the world, that are looking at drug targets. What goes wrong in the cell? What goes wrong in the cell first, second, third? What is most devastating? To identify which is the best drug target. So we've got a lot of activity going on to be really excited about. Okay. Okay. Let's see. I think I've got a couple more questions here. Whoops. Okay. So one of the questions we have, and we get this one fairly often, is the storage material, do we know for a fact that the storage material that builds up in the neurons, that's what's toxic to the brain cells, if you will? And the answer is yes and no. You know, we know it's a problem, we know it's not functioning right, but the beauty of TFAB and some of the science that we showed you today is that it not only prompts the cell to take care of its garbage disposal problems. Lysosomes, we gave you a very cursory overview, but lysosomes are responsible for many, many processes within the cell. And so if we increase their number, we're increasing the number of lysosomes that are capable of getting all of the work done. So on the one hand, it does remove the storage material. On the other hand, if there are byproducts, there are other things going on that aren't specifically directly connected to the potential toxicity of the storage material, then that's taking care of it as well. Next question. So are too many lysosomes a bad thing? And that's a really good question, because we always have to talk about, you know, when we talk about drug screening and determining what goes right in the cell, what goes wrong, and how to fix it, dosage comes into play. And so are too many lysosomes a bad thing? Is it bad if we put in too much TFAB or too many TFAB activators? At this point, we don't see that in the preliminary data. We don't see it as an issue. It hasn't happened in the animal model. But this is another reason why we have to be really, really careful in our preclinical studies to make sure that we get it right, so that the therapies that we develop are both effective and are safe for our children. Okay, so I don't have any other questions. Does anyone else have any other questions? We've got about 15 more minutes if you'd like.
In the meantime, if you have any other questions, after the webinar today, feel free to. Hopefully, you've got all the contact information there for Mary Beth, Danielle, or me. So certainly feel free to give us a call or shoot us an email anytime. And also, like we've said a couple of times, if there are other topics that that you'd like to see or hear about in future webinars, then we'd be happy to include those, as we would like to continue to create a higher level of communication with all the stakeholders and interested parties in the ongoing research development. And so we're open anytime, and certainly want to continue to have a transparent approach to this and provide as much information as possible. So like I said, we're here and happy to answer any questions at any time.
Absolutely. Mary Beth was right when she said at the beginning, no question is too big or too small. And please also let us know if these are convenient times for you. Last night and today, a convenient time for webinars. If the webinar format is something that works for you, or there's a different format, whether that's a conference call or in our newsletter. So please, let us know how we can be transparent and communicative.
Okay, great. Well, I guess we'll wrap it up. Thank you very much for attending, and we want to especially thank again all of our colleagues in family foundations and co-funding partners around the world for their support and their ongoing involvement, and certainly all their efforts to advance the cause for all of us. So we're grateful for that, and we look forward to continuing to work with you going forward. So hope everybody has a great day, and their summer's off to a great start, and we'll hopefully talk to you all soon, and we will certainly be at the BDSRA Family Conference in Nashville, and so if you're there, love to see you there. So have a great day, everyone.